This is part 2 of our DNA chapter, the first part being the lecture video about the scientists and all their discoveries that led to the structure of DNA. So today we're going to talk about the structure of DNA, but before we do that, we have to talk about James Watson and Francis Crick, who are the scientists that got the credit for discovering the structure of DNA, even though they actually weren't the people who did all the research behind finding the structure. The two scientists who really deserve all the credit are Rosalind Franklin and Maurice Wilkins, and they discovered the 2D model of DNA by using X-ray diffraction, and they discovered that there's a double helix. They discovered that there are nucleotides, but they never actually made the 3D model of DNA. And what Watson and Crick did is they took the X-ray diffraction and they took the 2D model that Rosalind Franklin and Maurice Wilkins made, and they made a 3D model out of it so people could understand exactly what's going on. And they actually, from that, were able to discover that not only were there two helixes, but they actually twisted like a spiral staircase. And they are the two scientists that got the credit for discovering the double helix structure of DNA. So as stated previously, DNA is what we call a double helix. Double meaning that there are two major strands on the outside, and helix meaning that it's twisted. So when we talk about the structure of DNA, we're going to delve a little deeper into what we call the subunits or the nucleotides that make up this double helix. So here we have our nucleotides, and a nucleotide is made up of three major parts. We have a phosphate group, we have one 5-carbon sugar, the name of it is called deoxyribose, and we have one of the 4 nitrogenous bases. And of those 4 bases, we have adenine, which is represented with an A, we have guanine represented with a G, thymine represented with a T, and cytosine, which is represented with a C. So within one nucleotide, we have one phosphate group, we have one sugar, which has 5 carbons on it, so it looks like a pentagon, and the name of that sugar is deoxyribose, and one of the 4 choices here from our nitrogenous bases. So we can have a phosphate, the sugar, and an adenine. That could be one nucleotide. We could have phosphate, a sugar, and a guanine. That can be a separate nucleotide, so on and so forth. Now the important thing here really is the sugar, because the sugar is what makes DNA different from RNA. That's one of the differences that they have between the two of them. And the term deoxyribose is in the name DNA. That's what DNA stands for, is deoxyribonucleic acid. And as we discussed before, out of the 4 macromolecules, carbs, lipids, proteins, and nucleic acids, DNA and RNA fall into that nucleic acid category. So that's where the name comes from. It's named after the sugar and the macromolecule group that it belongs to. So even though it's a really long name, once you break it down, it really makes sense as to why they named it that. Okay, so here's a picture of the nucleotide. We have our phosphate out here on the end. We have our deoxyribose sugar in between the phosphate and one of the 4 bases. Like I said, this could be an adenine, this could be a guanine, this could be a thymine, this could be a cytosine, but it's only one of the 4. The helix portion that I was talking about before, this outside, if we look over here at this picture, this outer portion, that's made up of repeating units of phosphate and sugars. That's all this is. Phosphate, sugar, phosphate, sugar, phosphate, sugar. The stuff on the inside, which we call the rungs of the ladder, right? If this were to be a ladder, a twisted ladder, the steps would be the bases. Now, because we have a set of nucleotides on one side and a set of nucleotides on the other, the bases connect in the middle, and that's how you have the base going across the entire middle section. When we talk about bonds in DNA, it's very important that we discuss the two different types of bonds that there are and their properties. So first, let's talk about the covalent bonds. As we've stated before earlier in the year, we said the covalent bonds are the strongest bonds that are possible. And these are the ones that are going to connect the phosphate to the sugar. So remember, as I said, we're going to compare DNA to a ladder. And if we were to have, or if we were to call this portion right here the handrails of the ladder, we would say that the handrails are made up of sugars and phosphates, and those sugars and phosphates are connected together by covalent bonds. Now that's super important because covalent bonds are very, very hard to break, and we do not want the handrails or the outer portion of the molecule to ever break. It's very important that this stays together, because if this portion were to break, then we would lose the sequence of the bases, and we don't want to do that. So we have the sugar and the phosphate, which are connected to covalent bonds, and the covalent bonds are very strong and they should never break. Now, in the in-between portion, so again, if we're talking about the DNA molecule being a ladder, the steps of the ladder, which is the inner portion, they are connected by hydrogen bonds. Hydrogen bonds are very weak. Now this is again very important because, like I said earlier, we have a set of nucleotides on one side and a set of nucleotides on the other in between. All right, so we have one base here and another base here. Connecting them is the weak hydrogen bonds. Now they're strong enough to hold this together to a point, but during DNA replication, during transcription and translation, which we'll get to talk about later on in the chapter, it's very important that we need these bases to separate. So if they were held together by covalent bonds, then DNA wouldn't be able to replicate, DNA wouldn't be able to turn into, well, not turn into, but to unwind in order for RNA to bond with it, to send messages, in order for transcription and translation to happen. So we need those weak bonds in the center, and those are the hydrogen bonds. As stated before, the hydrogen bonds help the bases, they hold them together, but it also allows for DNA to unzip or unwind, so replication, transcription, and translation can occur. So let's take a closer look at where these covalent and hydrogen bonds are located. So first of all, let's break this down. Up here we have our phosphate. Here we have our deoxyribose sugar, and here is one of the four bases. This one here is cytosine, represented with a C. Over here we have a separate nucleotide. Here's the phosphate here, here's the sugar, and here's our base. So with the phosphate and the sugar, remember I said this is what makes up the handrails of the ladder, is the sugar, phosphate, or phosphate, sugar, phosphate, sugar. This is our phosphate sugar bonds. And in between, what's bonding them together is the covalent strong bond. So we have our phosphate and our sugar making up the handrails, and in between there is our covalent bond. As I said before, what makes up the stair portion of our ladder is the connection of the two nucleotide bases. In between here, we have our hydrogen bond right here. Now we also do have another connection for the covalent bond. Not only do we have the connection of the phosphate to the sugar, but we also have the connection of the sugar to the base. So we actually have two sites of covalent bonding and one site of hydrogen bonding. Remember, the hydrogen bonds are weak, so we do want them to split apart at some point, but we do not want the sugar getting disconnected from the nucleotide, from the nitrogen base, and we also don't want the sugar and the phosphate getting disconnected from each other either. So that's why we have our covalent strong bonds here and our weak hydrogen bonds in the middle. So now that we've talked about the bonds, let's go into the nitrogenous bases. So, as stated before, the sugar and the phosphate, they make up the outer handrail portion of the ladder of our DNA molecule. And as long as we're talking about the molecule of DNA, the sugar will always be deoxyribose and the phosphate will always just be the phosphate group. And like I said, that's a repeating unit that will not change in the molecule of DNA. But as stated before, we do have 4 different nitrogenous bases. We have adenine and guanine, thymine, and cytosine. Now we're breaking them up like this because there are 2 different types of nitrogenous bases. We have a group called purines and a group called pyrimidines. The only difference is their structure and how many carbons they have. So adenine and guanine, their structure is made of a double ring, and thymine and cytosine are made up of single rings. So we would call adenine and guanine purines, thymine and cytosine pyrimidines. Now it's important to realize that purines and purines cannot bond together, and pyrimidines cannot bond together. So therefore, we have to bond one purine with one pyrimidine, but it can't switch around. It always has to be the same. And the reason why purines can't bond with each other is because they would have, let's see, so you have 2 per each, so that would be 4 rings connected, right, when they come together. And for the pyrimidines bonding together, you would only have 2 because they're single ringed each. And in order for the molecule to be even and symmetric on both sides, you would need to have one pyrimidine match with one purine in order to get a bond of 3 rings all together. So we always have adenine bonding with thymine. A always bonds with T. Adenine being a purine, thymine being a pyrimidine. And we always have cytosine bonding with guanine. So C will always bond with G. This never ever changes. With the molecule of DNA, if you see an adenine on the opposite side, we call these complementary base pairs. Complementary meaning the opposite side of the molecule. So if you see A on one side, you'll see a T on the other. If you see a T on one side, you'll see an A on the other. Same thing with cytosine and guanine. If you see a cytosine, a C, you will see a G on the other side, which also states that you'll see the same percentage of each within the molecule of DNA, because if you have an A on one side, you always have a T on the other, so therefore you'll always have the same percentage when you look at a particular genetic code. The person who came up with this is Chargaff, as stated in the notes that we took the other day. Chargaff's main discovery is the discovery of the complementary base pairs. He is the person that looked at the model and realized that there is the same percentage of A to T as there is for C to G, which is super important because this gives us a genetic code that we can follow, and it gives us the variety of different genetic code for different organisms. So as stated before, adenine will always bond with thymine, cytosine will always bond with guanine, and that is just the base pairing rules. We like to call them complementary base pairs. So if we look at this sequence here at the bottom, we're going to go through each one and we're going to complete the complementary base sequence. So when we talk about sequencing, this right here would be a DNA sequence, but this is only half of it. We have to find the complementary sequence, which it would be on the other side of the molecule. These lines here would be representing hydrogen bonds because it's bonding the 2 nitrogenous bases together. Remember, these are the weak bonds. So if we ever see a T for thymine, we will always see an A. If we see an A, we'll see a T. If we see a C, we'll see a G, G with C, and vice versa. So they're always going to match up with their other pair. Now there is an acronym that I always like to use, and that would be apple tree garbage can. Pick an apple from the tree, throw it in the garbage can. Apple for adenine, tree for thymine, garbage for guanine, and can for cytosine. I know it may be silly, but it always helps. So try to think of apple tree, AT, garbage can, GC.